두 번째
The second speaker is Professor Chang Hyeshik from Seoul National University. He's working for the School of the Biological Science. I would like to briefly introduce Dr. Chang to you. Dr. Chang graduated from the Yonsei University and he majored in the bio and brain engineering from the KAIST for the master's degree. And he got the PhD from the biological science of the SNU. He's currently working with the IBS RNA Research Institute, which is headed by Professor Kim. He's the research fellow for the IBS RNA. Starting from 2019, he started to teach at the Seoul National University, and he is currently associate professor of the Seoul National University. And he was working for the Dr. Kim's lab, and during his PhD course, he studied a lot about the RNA. And his research outcomes will be really interesting, which is about the transcription and the transcriptome about the SARS-CoV-2 that has been published on the Cell. So that transcriptome was found, which means it's a really significant milestone because that can be the foundation for the exact diagnosis and the vaccine and the treatment development. Dr. Chang today will introduce us about the molecular biology of the coronavirus. Probably you have heard and read lots about the coronavirus. And he's going to tell us about the structure and the genetic information about the coronavirus. And also, he's going to tell us about the wonderful information about the life cycle of the coronavirus. Without further ado, I would like to invite Dr. Chang. Hello, my name is Chang Hyeshik, who is currently working for the School of the Biological Science. I'm in charge of the transcriptome team in the IBS RNA Research Institute. I'm currently researching the RNA from the coronavirus. Today I'm going to tell you about how it looks like, how it is composed of, what it works. And from the molecular biological perspective, I'm going to deliver the scientific facts about the coronavirus in a casual way. As you may understand, the life of last year and the life of this year is totally changed. For example, you need to have the social distancing while you are queuing up. And on the street, almost everyone is wearing the face mask. Last year, you didn't even imagine that you're gonna eat together with the transparent plastic in between the two. And working at home was confined to a certain industry, but everyone is working remotely. For the remote conference in the past, not many people know about it. However, just with a simple address, everybody can have access to the remote conference. All the people are getting used to the online lecture and the remote lecture as well. The impact of the COVID-19 is so big, so there is an unprecedented effort of the scientists across the world. And I would say it accounts for half of all the research tasks of the scientific field. It's more like a human alliance against the alien. So the scientific avengers are trying to defend ourselves against the alien. And many people bring their research methodology, technique, and the research interest so that people can make a concerted effort to build knowledge about COVID-19. When the alien comes in, you may be wondering how it looks like. And the pandemic is not likely to over shortly. Maybe it can last for the next a few years. When we are under attack, we need to understand who they are and what they are. For example, what are they eating? How do they look? And how many legs and the arms? What language do they use? How they give birth to? And how they get raised? So this is the knowledge that we needed to get prepared with rather than just being attacked by the virus. So let me tell you first how the coronavirus was found. It was first found in poultry. In the 1930s, within a poultry farm, chicks started to die en masse. 90% of them died. And the common factor was they have difficulty in breathing and they had a pneumonia. People wondered whether it is because of the germ or the virus. And then they found out it was because of the IBB, the virus in the 1950s. There is a biological lab located in London and the mice died en masse. So they studied a little bit more and many of them had hepatitis. And the researchers found the liver had lots of virus in it. This is called MHB, and this research has been widely used even now. We found coronavirus in the human bodies in the 1960s. UK NHS established an influenza research institute in 1945, and they looked into what was the cause for the flu. There were some known viruses, of course, and they removed those known viruses and then they found a new virus strain called B814, naming after the sample. And then they continued the research and then they found a new strain 229E and the OC43. Soon after, electron microscope was discovered and then they studied. Now, the famous crown shape was observed, and people started to call them as the coronavirus. Later, they found the IBB and MHB have the same shape. So, people grouped them and called them as a coronavirus. The coronavirus family started to threaten human lives in the 2000. The SARS coronavirus became prevalent in the early 2000, and people came to understand that coronavirus can threaten the human lives. And then HK1 was found in Hong Kong and NL63 was found in the Netherlands. They are not causing serious symptoms. And the MERS coronavirus, which contributed to the K quarantine, was found in 2010, and they become prevalent across the Middle East. And as you know, starting from last December, SARS coronavirus 2 has become prevalent as well. It is a pandemic, and coronavirus can threaten the human lives, and also it can give lots of the financial damage to us. It is transmittable to human, and we have 7 viruses. If we include the B814, we have 8 human transmissible viruses. However, we do not have B814 anymore. We do not have any sample for the comparison. So we can say we have 7 human transmissible coronaviruses. And probably we need to look into the evolutionary relations among them. And the coronavirus can be divided into alpha, beta, gamma, delta. And the alpha and beta can be human transmittable. Among the alpha virus, NL63 and 229E are human transmittable. They are only causing light cold, and the beta coronaviruses can be subdivided. And to tell you, sarbecovirus where SARS belongs, and morbecovirus where the MERS belongs. You can see the SARS-CoV-2 viruses. They've been sampled in the initial phase, so they code as Wuhan. And then they are most relevant to the SARS-CoV-1 among the human viruses. However, they are most relevant to the RATG13 if all the species are included. Probably they have the same ancestor. And the pangolin is further from it than that of the bad coronavirus. Let's take a look at the structure. It is a crown structure, and you can see the S protein or the spike protein. This binds to the ACE2 protein of the human epithelial cell. It recognizes the human cell and gets the virus into the human cell. And there is a genomic RNA. And there are genes within the genomic RNA. And these genes will be translated into protein. And they make the outer structure and sometimes towards the harmful damage to the humans. RNA without protection is dangerous. So they needed to be protected. So there is a nucleocapsid which protects RNA. And also there is a phospholipid layer, as you can see. And if it is covered with this layer, it can stay stable. However, the cover or the layer is vulnerable to ethanol, so the layer can be broken with the alcohol sanitation, and even RNA is likely to be broken. And there are 2 types of the protein on that layer, the membrane protein and the envelope protein, and they make the structure for the virus and keep the structure stable. I would like to tell you about the life cycle about the virus. And you can see the inside the cell and you can see outside the cell. And there is a virus. The virus has the spikes at the outside of the virus, and then it can bind to the ACE2 protein of the epithelial cell of the lung or the airway. And the ACE2 is not friendly to the coronavirus. Actually, they are modulating the blood pressure. However, coronavirus is taking advantage of the ACE2 and they simply get into the human cell. Once it gets into the human body, the cover will be gone and then the protected RNA comes in. It is like a straight line and it has a cap on the front and it has 8 tails at the back. Cap and 8 tails. And the long ones are similar to the messenger RNA. They can naturally generate a protein. And from NSP to the NSP16, they make 16 proteins. When these 16 proteins are made, the gRNA wasn't able to do anything, however, but they make a reversed version of the copy. And in the case of the DNA RNA, they should be matching so that they can be copied. And which means when a new pair is made, the direction of the new one is reversed from the original one. Ace match U is generated. For the old one, this was the back, and for the new one, this is the front. So it is reversed. So this is why we'll call them as a minus strand subgenomic RNA. And I'm going to tell you more about the length changes. And once it is copied, it should come back to the original direction. So they create the plus strand subgenomic RNA. And within this, there is ORF. And within the RNA, they can contribute to the protein generation. They will be translated into the protein like SEMN and other types of the protein. They are ready to become more viral. And so that they can have some infectious viral compounds. And what is really important is gRNA is missing here. To create the gRNA, they make a copy once again, which is the opposite direction of the original. So they make the minus strand genomic RNA out of the plus strand genomic RNA. And then repeat this process again to create the plus strand. This will be covered by the N protein because RNA cannot move around unprotected. So this goes out of the cell, so that it can attack other cells outside of the human body. And by doing so, the coronavirus can be proliferated and then they can infect other cells. And now, let's take a look at what is inside the gRNA. And on the line number 3, you can see the structure of the SARS-CoV virus. And the SARS coronavirus and the SARS coronavirus 2 is almost the same. And on the first half, you can see the ORF-A 1A and 1B. And then they have the key protein that needs to be generated and they are encrypted in this area. So they read this information because they are like a blueprint. So you can see the structural protein and the accessory protein. And the alpha coronavirus and the beta coronavirus have the first half, and that is almost the same. So the first half is very similar and they are preserved. However, the second half is not well preserved, and because they are not similar. However, the spike EMN protein are the core proteins. The spike on the outside, the N, the cover virus, and the M and E on the layer are the core proteins. So they will be preserved regardless of the strain. And now let's take a look at how the RNA will be generated. I told you about the ORF1A and 1B, and RNA is in the cell, and in that case, from the first or the lead ORF, proteins can be generated. The first one is ORF1A and 1B, and they are generated at once from the NSP1 to NSP11, and they are related to ORF1A. And for the ORF1B, as they are coming from the beginning, 1A and 1B come together, and then the protein 1 to 16 will be generated. There are some overlapping parts, but why did they give the different name? Because there is a frame shift in the middle. Sometimes it is used, sometimes it is not used. I will give you further explanation about that at the later part of this presentation. The long one was made, and then in this case, they needed to be broken down. I will give you an analogy. When you are building a house with the wood, you have the trunk and the branches, and the short ones will be used for the furniture and then structures. So the further breakdown is really necessary so that you can make 11 proteins as an outcome of the breaking down, or 15 proteins. And in that case, the number 11 is gone, and then the 11 is prolonged, and then it becomes the number 12. This is the protease, the breaking down factor. And once the protease is inhibited, there is no further protein breakdown. And then in this case, the protein bundle is moving around and it cannot work properly. And by doing so, we can protect the viral actions. Sometimes the long ones and the short one coming on, and I'm going to tell you about the mechanisms.
The RNA consists of 4. However, the protein has 20 starting from A to Z. The 4 cannot code 20 types. So the combination of the 4 plus 4 and 4 is used to code 20 types. Let me show you what it is like. It's much like how we build each syllable and words using the alphabet, in this case, Korean alphabet. Let's consider these are proteins. In Korean, we can write here 밥 먹음 끝, which means I'm done with the meal. As you see here, each letter consists of 2 consonants and 1 vowel. A consonant is followed by a vowel and another consonant. First consonant followed by a vowel and the final consonant. It is like a syllable consisting of RNA 3 letters. When the translation of the protein is completed, here we have this one. Here we have I'm done with the meal. However, if there's a frame shift, the last vowel in the final letter is prolonged to push the final consonant into the next spot. So here are the consequences. The last letter is switched into a syllable, missing the final consonant. This affects the subsequent syllable. When there is a frame shifting taking place, that is called the read-through, there comes the long one consisting of 16, and then the outcome without the frame shift is the shorter one consisting of 11. And this is the mechanism of -1 frame shift, meaning that the next syllable begins with the final consonant pushed down from the previous syllable. Why does this happen? Let's explain. RNA is like a necklace whose component is connected with magnet in between. Sometimes a certain part of RNA is formed by being twisted among themselves. This is why frame shifting is taking place to end the word with a vowel, and the final consonant is slid into the next syllable. This mechanism is quite interesting. Let me have a look at this area. In the human cell, only the front ORF section is turned into protein. This is an ending at the end of ORF1A and another ending at ORF1B. It is impossible to make this protein going through the front section or the through. So something different needs to take place in order to make 3A the cap. So here we have the lead-up created, which does not have the front part. And this is called the subgenomic RNA. As you see, the front section is skipped to make S and 3A. This is how the subgenomic RNA is created. This is how the variety of a protein is created. This single genome RNA can generate a big number of proteins ranging from 9 to 13. There are different definitions, and they also generate many more diverse ones. So some others may think differently from me. Let's look at the details. This is genome RNA of COVID virus. Here we have the sequence named body TRS. And then at front, we have another TRS right next to the leader. As there are 2 TRS, so sometimes there can be a confusion as to where to go. So there comes a shorter one, as these sections could be hopped over or skipped. And we have shorter ones, and sometimes we have longer ones. As this is a minus strand, it is made into plus a strand to finally make proteins. So as I said, we have shorter ones or longer ones going through this process. So this is the minus strand, and it is made into the plus strand to make the final protein, as I said. When it comes to genome, it cannot be copied exactly. This means that they have to create the minus in order to make plus in the end. So during this process of generating genome RNA, this kind of recombination occurs from time to time. Most of them are shorter ones, and they do not contain all the things that are necessary for virus. So they are not likely to have infectivity when they enter the cells. But if a cell is infected by several COVID viruses at the same time, then recombination may occur. Let me have a look at the protein one by one. NSP3 and 5 are quite interesting ones. They are protease. As mentioned earlier, they are enzymes to cut proteins. It looks like enzymes found in a papaya called the papain, so it is named papain-like protease. Here we have a 3C-like protease, and it looks like a 3C, an enzyme found in other virus. Without an enzyme to cut the protein, they cannot properly build proteins. This means that they are very important to virus and become target of treatment. Here, NSP3 is RNA-dependent RNA polymerase engaged in the replication. Without NSP12, no replication can take place. Since RDRP is of great importance to the virus, there is much less chance of having its mutation. Even those COVID viruses that are distantly related, then they tend to have a similarity in RDRP. For those viruses, distantly related ones with accumulated mutations, one may not able to detect the virus. However, we can reduce such diagnosis failure by targeting RDRP. These are NSP7 and 8, enzymes called primase. They assist RNA replication. To be more specific, they help RDRP for RNA replication. RDRP needs primer to begin a replication process, so NSP7 produces the primer and helps with the help of NSP8. Then comes NSP13. I mentioned that RNA is like a magnetic necklace, highly likely to get stuck each other. Once they get stuck, it makes quite difficult to copy. This can be resolved in the helicase. Then comes NSP14, an exonuclease. COVID virus is as long as around 30,000 letters. It is exceptionally big for an RNA virus. Actually, it is the biggest among RNA viruses. RNA virus replication is not precise enough. This means that there could be errors accumulating. A little bit of errors do not matter, but too many of them disrupt the infectivity of the virus. So exonuclease is introduced to erase those errors. It is like a monkey typing. Let a monkey do typing and then press the backspace whenever the monkey pushes a wrong key. Here, the errors are deleted with NSP14. Then comes NSP16, which is methyltransferase. It attaches the group made of a carbon and 3 hydrogens. For RNA regeneration, it is better strategy for virus to create the RNA the same as the one found in the human cell messenger RNA, as it's much more stable and better at producing protein. So this is called MG7, which refers to the gRNA with methyl added on the front. This is called capping, as it looks like wearing a cap. It helps make the RNA, virus RNA, exactly the same as a human RNA. Let me now cover those who control the host. One of the best known is NSP1. NSP1 blocks the translation of the host cell. This means that the protein is not produced anymore in the host cell if NSP blocks the translation. It prevents the host from launching response to the virus. It also eliminates the mRNA, which is used to produce protein. As there is no mRNA, the host does not have the drawing for protein production, which robs the host of the ability to launch a response. In the end, more than half of the mRNA of the host cells will be replaced with the virus, the mRNA, which turns the host into the factory producing the viruses. This leads to the cell cycle arrest. When virus enters the human body, it triggers the innate immune system. To be more specific, the interferon signaling is turned on. This protein blocks the interferon signaling. In a nutshell, NSP1 plays an important role to allow the virus to occupy the host. NSP3 removes labels. In order to remove the invader protein, the host puts a label to them so that they can be removed in the end. Upon the detection of the virus, the host tries to come into the midst of the war and produce weapons by eliminating the existing ones used in time of peace. However, this cannot be done because of NSP3 blocks this. They remove all the ubiquitin labels, and the host cells are not able to do the things that are necessary, that are supposed to do. And there are oral RF3A and 7A, and 7A helps the virus exit the cell. And they make holes in the cell membrane and induce apoptosis and causes inflammation. So there are those ones that help the virus to escape once everything is done. As seen, this virus is armed with very good weapons. But this does not mean that they are the main reasons of the successful domination of the world by SARS-CoV-2. Actually, other viruses are equipped with similar weapons. And some factors are coincided to have a high infectivity in humans. Scientists have studied the life cycle of virus in order to understand them better. There are many different steps that the virus goes through inside the body. The entry of the virus can be blocked by chemostat, Abidor, and chloroquine, and hydrochloroquine was well known to the public as it's championed by President Trump. But its clinical study proved that it does not have a good efficacy. The next step is blocking the cutting of the protein produced by the virus. This pathway is widely targeted to treat the infection of other viruses. One of the good examples is lopinavir, and we have many more candidates. We have other class of treatment like remdesivir, which inhibits the RNA synthesis. Let me briefly explain the MOA. RNA consists of 4 letters. And let's assume that we have cards, each of which having a letter out of the 4 letters. Those letters are combined to do replication. And let's say that we have a letter card G in addition to C. As C and G look alike, G may be used instead of C from time to time. This means that there are errors accumulated blocking RDRP from properly fulfilling its mission of RNA replication. There are many other antiviral candidates currently under study, and the scientists will develop medications with new targets. And it takes a long time to develop a new drug. Usually it can take 10 or more years. But it seems that this pandemic may last longer than 1 or 2 years. And as we continue to do our research on the virus, we will have a better understanding of COVID virus. And this will stimulate the development of a new antivirus. And on a long-term perspective, what scientists have learned can be used for the future. And this let many of us continue to do the study of the COVID viruses. And let me give you my last comment. And this virus is armed with so many things. However, they cannot do anything unless they come into the host, our body. Virus does not have any wireless communication with the cells of our body, or they cannot fire laser weapons. So the best strategy is not to allow them to enter. This means that we have to wear masks and use sanitizers and keep doing our best, complying with the best practices. Thank you.
Thank you very much for the detailed and the kind explanation. I would like to thank Dr. Chang. Once again, thank you very much, Professor Chang. And his research was published in the Cell, and he created almost perfect the gene map and the RNA map. And as he said, and I sincerely hope we can have the exact and accurate diagnosis system and to develop the effective antiviral drugs. And once again, I would like to thank Dr. Chang for the wonderful explanation. And he explained us from the molecular biological perspective. He gave us a detailed explanation about the structure, about the virus. Thank you.